Good day, everyone. So for today, we're going to discuss about hemoglobin synthesis. So in the picture, you can see an RBC, and inside the RBC, it's actually our membrane, RBC membrane, and within that RBC, you can find bags of hemoglobin. And of course, the function of our hemoglobin is to carry our oxygen throughout the body. So the two functional cellular constituents of our RBCs are the following. So we have the membrane, the RBC membrane, and the other one is the hemoglobin molecule. So our hemoglobin is actually approximately 34 grams per dl in its concentration within the RBC. It is actually the value of our mean corpuscular hemoglobin concentration. Okay, so our molecular weight, the molecular weight of hemoglobin is 64,000 daltons, and its main function is to transport oxygen, okay, from the lungs and deliver it throughout the body. Okay, so your hemoglobin constitutes 33% of RBC's weight by volume. Hemoglobin is a conjugated protein of heme and globin. It consists of 4 heme groups and 2 pairs of unlike polypeptide chains. Okay, so in the picture, as you can see, this is a picture of a hemoglobin, an adult hemoglobin. So an adult hemoglobin consists of um, 2 beta and 2 alpha, or 2 pairs of unlike polypeptide chains. So the main component of our RBCs, okay, these are the main component of our RBCs, the heme, the heme and the globin, and of course in each heme you can find there our iron, which is in ferrous form, or Fe2+. So if our hemoglobin is free, or free hemoglobin. So by the way, free hemoglobin is a hemoglobin that doesn't have membrane anymore, and that free hemoglobin, okay, does not survive for long. Okay, it doesn't last until 120 days like your ordinary RBC. Okay, so since it is free hemoglobin, it is very exposed to the environment, outside environment. So eventually it will be rapidly salvaged and excreted. So another picture of our hemoglobin molecule. Okay, so each of the 4 globins uh, has a heme, and within or in the center of the heme you can find the ferrous form of iron. And our heme is actually consists of ring of carbon, hydrogen, and nitrogen. So hemoglobin synthesis, the making of your hemoglobin, depends on the following. It depends on the adequate supply and delivery of iron, because of course in each heme you need one iron also to be attached within one heme. And another one is adequate synthesis of protoporphyrins, since protoporphyrins are the precursor, or yeah, the precursor of your heme. Okay, so if there is adequate supply of protoporphyrins, then you will have adequate also of supply of heme. And then you need also adequate globin synthesis. Okay, so very important thing, you need to have a balance between your supply of protoporphyrin and globin. Okay, because if globin is lacking or inadequate, okay, more protoporphyrins will not have a pair. Okay, so at the same way, if your protoporphyrin is lacking, more globin will have um, no pair, no pair as well. So of course it has to be balanced so that it can create our molecule of hemoglobin. So if it's not balanced, there could be a disorder that is related to our hemoglobin, or it is known as hemoglobinopathies. So hemoglobinopathies are actually um, disorders that is related to heme, and one of those um, hemoglobinopathies is caused by excessive number of globin and inadequate number of protoporphyrin, or it's either excessive synthesis of protoporphyrin but inadequate uh, supply of globin. So not balanced at all. So if in that case, if it's not balanced, then there could be hemoglobinopathy that will happen to the patient. So heme. Okay, so heme is again consists of a ring of carbon, hydrogen, and nitrogen atoms. It is called protoporphyrin 9 with an atom in the center, and that atom is in ferrous form. So protoporphyrin 9 is attached with an iron, a ferrous form of iron, and when it is attached with an iron, then that is already called a heme. A heme is also known as ferroprotoporphyrin. Okay. Heme group is positioned in the pocket of polypeptide chains, and one heme molecule can combine reversibly with one molecule of oxygen. So there are four heme groups in one molecule of hemoglobin. So therefore, in one molecule of hemoglobin, it is capable of carrying four molecules of oxygen. And heme, by the way, is the one that is responsible for the color of our blood, which is red. Okay, so again, the picture of the hemoglobin molecule, you have there four globins, and then within the globin, there's a pocket where your heme is suspended or located, and within each of those hemes, in the center of those heme, you can find the iron, which is in ferrous form. So iron in ferrous form in the center. So for globin, it is consists of two pairs of polypeptide chains. So for example, in the picture, you have an adult hemoglobin there. So an adult hemoglobin is composed of two beta and two alpha chains. Okay, two pairs of unlike polypeptide chains. So each chain of our globins, because aside from alpha and beta, you also have your gamma, your delta, your zeta, your epsilon. The difference between those chains, Greek letter chains, are the number of amino acids, or even the variation of amino acids in your globin. Okay, so per globin, okay, it has polypeptide chains, or it is divided into 8 helices, okay, from A to H. So 1, A, B, C, D, E, F, G, H. So 8 helices. So again, as I've said, differences of those Greek letter globins is due to the variation of amino acid sequences, or even the number of amino acids. So this is a picture of your globin. Okay, as you can see, you can find the 8 helices: A, B, C, or D, E, F, G. Okay, and in the pocket, okay, in the pocket, it is located. It is the place where your heme is being suspended or located. It is actually between your E and F helices. So your hemoglobin is being synthesized during the nucleated stages until in the reticulocyte stage. In fact, 65% of synthesis of this hemoglobin, okay, is occurring during the nucleated stages, and the other 35% occurs during the um, reticulocyte stage. Okay, so again, 65% of synthesis of your hemoglobin occurs during the nucleated stages, and the other 35% occurs during the reticulocyte stage. So your hemoglobin molecule is actually being characterized into four structures: the primary, secondary, tertiary, and quaternary. For primary structure, this refers to the arrangement of amino acids in the polypeptide chains. So as you can see in the picture, you have a lot of amino acids. They are arranged in a polypeptide chains. So another primary structure of your globin. So secondary structure is composed of polypeptide chains arranged in helices and non-helical segments. Okay, so you have the helices, the 8 helices from A to H, and of course the non-helices, so the C terminus, the N terminus. Okay, and it is also composed of, again, non-helical segments, alpha plated and the beta plated, or yeah. Tertiary structure. Tertiary structure is composed of arrangement of helices into three-dimensional structure, and globin chains are looped to form a pocket for heme, and within that pocket, okay, there lies the heme. And within the heme, you can find an atom, an iron atom at the center of that heme molecule. Okay, so the heme group is suspended between E and the F helices. For the quaternary structure, this is the complete molecule of hemoglobin. It is spherical shaped and it has 4 heme groups attached to 4 polypeptide chains. So 1 heme per 1 globin. It can carry 4 molecules of oxygen since there are 4 globins, and each of those globins is attached with 1 heme. So therefore, 4 hemes, then 4 oxygen will be carried by 1 molecule of hemoglobin. So again, a quaternary structure of your complete molecule of hemoglobin. So there you have your primary structure composed of sequence of your amino acids, secondary structure composed of alpha helices and beta sheets, and then tertiary structure. This time this is already spherical in nature, and then quaternary structure. This is the whole, or this is a complete structure of your hemoglobin. So heme synthesis occurs in the bone marrow. Okay, first in the mitochondria, and it will be transferred to cytoplasm of your erythrocyte precursor, and then it goes back to the mitochondria, especially in the last stage of synthesizing your hemoglobin. So your mature RBC cannot synthesize any more hemoglobin because it doesn't have mitochondria anymore. Okay, so protoporphyrin synthesis occurs both in mitochondria. It will transfer to cytoplasm and it goes back to the mitochondria. So just like in this picture, this is a sequence of your heme synthesis. Okay, so from succinyl-CoA, it will bind to glycine, and the enzyme ALA synthase will help your succinyl-CoA and glycine to become aminolavulinic acid, or the ALA. It happens in the mitochondria, and after that, okay, the synthesis will happen in the cytoplasm. Okay, so your ALA, okay, with the help of your ALA dehydratase, okay, it will become porphobilinogen. Your porphobilinogen, okay, with the help of your hydroxybilane synthase, okay, your porphobilinogen will become hydroxymethylbilane. Your hydroxymethylbilane will be helped by your UPG3 synthase. That's an enzyme that will help your hydroxymethylbilane to become uroporphyrinogen 3. Your uroporphyrinogen 3, okay, with the help of your UPG decarboxylase, will help your uroporphyrinogen 3 to become corproporphyrinogen 3. Your coproporphyrinogen 3, with the help of your CpG oxidase, will become protoporphyrinogen 9. So as you can see, uh, the orange, or the orange color here, signifies that the synthesis of hemoglobin goes back to the mitochondria already. So this one, it happens in the mitochondria, and then it happens in the cytoplasm of your RBC precursor, and then it goes back to the it goes back to the mitochondria again. Okay, so so your protoporphyrinogen 9, with the help of your PPG oxidase, okay, will become protoporphyrin 9, which is your precursor of your heme. Okay, so these narrations are actually the ones that I have explained in the process of synthesizing heme. Okay, so you can read those narrations at home as you review this video. Okay, so this is also another picture of how your heme is being synthesized. So this is the final touch. Okay, final heme production. Okay, your precursor of heme, your protoporphyrinogen porphyrin 9, okay, in the mitochondria will be binded with your iron, your ferrous form of iron, which is transported by your transferrin. With the help of your ferrochelatase, it will then become heme. Okay, so once protoporphyrin 9 and iron combines together, okay, that combination is already the heme. And all of this is the last part, this happens in the mitochondria. So now let's talk about your iron. Iron is delivered to the membrane of your RBC precursor by their protein carrier transferrin. So transferrin is the one that transfers or delivers your iron, okay, from the source, okay, from the storage of your iron in the body to the membrane of your RBC precursor. Okay, your iron here is committed to hemoglobin synthesis. Okay, and it proceeds to the mitochondria to be added to the protoporphyrin ring to form a heme. So again, the iron form here is the ferrous form. So 2/3 of the total body iron supply is bound to heme in the hemoglobin. Okay, so the other 1/3 is going to be placed or stored, and it could also be released, okay, especially for females. Once they have their monthly period, it would also be released, but more of those 1/3 of your iron are being stored. So globin synthesis. Okay, so this time let's talk about globin. So again, globin consists of 2 pairs of polypeptide chains. A total of 4 globin chain is in your 1 hemoglobin molecule, just like in that picture. Okay, it is composed of simple proteins, consists only of amino acids, which are connected by peptide bonds. And your different globins, like your alpha, beta, gamma, zeta, epsilon, okay, those Greek letter names of your globins, they're different uh, because of the amino sequences in their within it. So those globins I've mentioned, they are comprised of 141 to 146 amino acids. Okay, so as I've said, those polypeptide chains, your globins, they are different because of the variation in the amino acid sequences. Okay, so who controls your polypeptide chains or your globins? Your chromosome 16 and 11. Okay, so each of those globins or each of those polypeptide chains is divided into 8 helices from A to H. Now, alpha and zeta genes are alpha and zeta are actually alpha globin genes. Okay, so the alpha and the zeta, these two are actually controlled by chromosome 16. And the others, okay, the epsilon, the gamma, the delta, and the beta, they are controlled by your chromosome 11. Okay, so here are the different globin chains or polypeptide chains. You have the alpha, beta, gamma A, gamma G, delta, epsilon, and zeta. Okay, alpha has 141 amino acids, and then beta has 146, gamma A has 146 as well, but in the position 136, it is alanine. Okay, the amino acid there in the 136 position is alanine. However, in gamma G, in the position 136, the difference between gamma A and gamma G is the amino acid that is located in the position 136. Okay, so gamma A's amino acid in the position 136 is alanine, and gamma G's amino acid in the position 136 is lysine. Okay, for delta, it has 146 amino acids, epsilon has 146 as well, and zeta has 141. So as you can see, alpha and zeta has 146, and then the rest, okay, have a 146. Okay, the rest has 146, and then the alpha and the zeta has 141. So I've said this already, chromosome 16 is the one that controls your alpha and the zeta chain genes. Your chromosome 11 is the one that is controlling your gamma, beta, delta, epsilon chain genes. Okay, so your alpha, that's why there's two alpha, alpha 1, alpha 2, gamma A and gamma G. Okay, have you noticed that? There are two because these two have two copies of genes also. Two copies of gene per chromatid, so a total of 4. For your zeta, beta, delta, and epsilon, it only has one copy of the gene per chromatid, so a total of 2 genes. So the production of globin chains takes place from the pronormoblast to the reticulocyte stage. And globin proteins are made via transcription in the nucleus and translation in the ribosome. So when synthesized, the globin chains are released from the ribosomes. So finally, after translation in the ribosome, your globins will be released from the ribosome to the cytoplasm, and then the heme will also be released or will be completed. So these two will then combine and become hemoglobin. So as I've said, transcription happens in the nucleus, and when it goes out, it goes to the cytoplasm to be translated in the ribosome. And then after that, this will come out, and it will pair to a heme. So it's already hemoglobin. So I think that's all for today's session. So next week I will continue the discussion of the part 2 of our hemoglobin synthesis topic. Thank you so much for listening.